Welcome to Mendelian Inheritance. This is Anthropology Laboratory, Lesson 3. I am your professor, Nehemiah Rodriguez. So let's get started. Gregory Mendel. Let's call him a private scientist. He is known as the father of modern-day genetics. Because in 1965, he actually discovered the principles of inheritance while working at a monastery. He was an Australian monk. Now, his work that he did with his peas, with the pea plants, which we're going to go over, he ended up publishing. And his experiments on plant hybridization in 1867, roughly, he wrote it 60. So right after he discovered it, he wrote a paper and published it, but it was too contradictory at the time for popular beliefs, because what we're gonna explain is it went against this kind of blending hypothesis theory. So it got swept under the rug very quickly, and then no one knew about it for about 30 years. And it wasn't decades until it was finally appreciated. So what Mendel came up with was his laws of inheritance. Mendel's work demonstrated that parents pass on to their offspring discrete heritable factors, which we call genes nowadays, which retain their individuality generation after generation. Now, if they retain their individuality, then they're not really blending. So that's in contrast to the blending theory, which originally stated that traits just melted together. So here's what Mendel's experiments look like. So why would he choose peas? Well, when in doubt, keep it simple, right? So peas are easy to grow, and they were available in many distinguishable varieties. And you could control the reproductive output. This was the genius of Mendel. So you see, he can get a round seed form that's wrinkled. You can get a yellow seed form that's got a smooth texture. You know, the pods can have different, it can be inflated, constricted, all this stuff, different color flowers. So he was able to mix and match. So Mendel's peas experiments. So let's look at two of his true breeding lines. If Mendel breeded green with green, well, he always had green peas. If he breeded yellow with yellow, he always had yellow peas. Now let's start really experimenting, right? So let's cross them. Let's go with the generation F0, which we're going to call the parental generation, where you take a yellow and a green. Let's see what happens. After he did that, he found the first generation. All of the generation were yellow. So, in contrast to popular science at the time, if the blending hypothesis was correct, what would we expect from generation 1? You mix a green with a yellow. Well, I'm sure you've mixed paint before. You're going to expect a greenish yellow color. However, that wasn't the case. There's no greenish yellow peas. There's either green or yellow. And it turns out yellow peas are dominant. And we'll go over that later. But green peas are more popular. Not sure why. So Mendel learned that the blending inheritance was wrong, and one color was dominant. So he began to coin these terms. Okay, let's take it a step further. Mendel took the first generation that were yellow, and he bred those together, and he ended up getting a 3-to-1 ratio of green to yellow, where one fourth was always green, and the other 3 were always yellow. This is how he discovered that the yellow peas were dominant, because they were more frequent in the gene pool, in the phenotype as well. So Mendel inferred 2 laws. Independent segregation. characteristics of organisms are determined jointly by two particles, what we call genes, one inherited by each parent. There's a 1/2 probability of inheritance. However, he also had a second law of independent assortment, meaning the particles, or genes, for different traits are independently inherited, which is very important to pass down genes, right? You don't want them to blend. You want them to pass down as they are, and perhaps just a little altered. So Mendel's particles were genes on a chromosome. So remember Mendel's inferences. Characteristics of organisms are determined jointly by two particles, one inherited by each parent. That is independent segregation. So let's look at alleles. An allele is a variation of the same sequence of nucleotides at the same place in a long DNA molecule. Remember the nucleotides we went over in the last lesson, the A's, T's, C's, and G's, and then a pair makes a gene, and then a lot of them, a sequence also makes a gene, which makes the genetic code, right? So if we look here on a chromosome, we could see the gene for eye color, temperature regulation, gene for nails, and all that. So these are alleles, okay? These patches of genes are alleles. And we can see here allele A and allele A, lower case for the green, P, because these are for P's, is going to be recessive, and then the yellow we're going to represent by an uppercase A is going to be dominant. So if we're thinking about P's, if we're thinking about people, everyone has 2 copies of every gene called an allele. One comes from the mother and one comes from the father. So if you have a gene for an eye, well, obviously your mother's going to have a gene they're going to pass on for an eye, and your father's going to pass on a gene for an eye, not just one, unless there's a major mutation during the embryonic gestation or crossover events. So alleles. For the exercises that we're going to be doing are Punnett squares. The dominant, again, we're going to represent by an uppercase letter. The recessive will be a lowercase letter. And the 2 combined alleles is going to make up the genotype. So let's revisit genotypes, okay, and apply it specifically to this exercise, to this lab. And we're going to talk about homozygous and heterozygous. 1 or 2 dominant alleles present phenotypically will physically appear, okay? 2 recessive genes need to be present in order for the phenotype to be expressed. So what this means is frequency of dominant and recessive is important. So if we look here, we can see there's only 2 options for homozygote genotypes. TT, homo meaning same, hetero meaning different. You're going to hear that a lot in anthropology and in many sciences is, heterogeneity, heterozygosity, heterotypically, and homo, sapiens and homozygosity and homologous. All it means is if you see HOMO in the front of the word, that means that they're similar. Hetero means there is a variation. So let's look at this phenotype. Let's look at a homozygous versus heterozygous. But when we're talking about phenotypes, remember, that frequency is a factor and dominant alleles is a factor and dominant genes is a factor. So any homozygous dominant trait will always be expressed. Any heterozygous trait will always be expressed because there's only two options, heterozygous or homozygous. So if it's heterozygous, there's going to be a dominant gene there. But a homozygous recessive needs to be homozygous in order to be expressed. So there's less of a likelihood of a recessive gene to be expressed. We can see right here a 2-to-1 ratio.